Hello, soil conservation and management students at the Royal University of Agriculture. This is session 15 lecture series. This is video lecture number 2 on zone and grid soil sampling. Session 15 is on the application of GNSS and GIS in agriculture. I am Dr. Jared Williams with Brigham Young University, Idaho. In previous lecture, we introduced you to the applications of GNS and GIS in agriculture. We got to put that in there. In this lecture, we're going to focus on zone and grid soil sampling, which is a type of application of GNSS and GIS in agriculture. First, let's define soil sampling and the reasons why we would soil sample. Soil sampling is the idea that we can measure the level of nutrients in the field and therefore make decisions on how much additional nutrient, whether that's through compost or manure or fertilizers, needs to be added to meet crop demand and our yield goal. We often pull soil samples and measure the amount of nitrogen, phosphorus, potassium, and other nutrients, as well as determining the level of soil pH to determine whether we need to apply lime or modify our fertilizer management practice based on the soil pH. Most soil sampling programs, or what we will refer to as traditional soil sampling, is where we take multiple points or multiple samples within the field at random locations. As you can see on this map, we've sampled in random locations. We then take each of those individual soil samples and we composite those into one single soil sample. When we call that a composite or bulk sample. But we've essentially taken each of these individual soil samples from the field and we have put them into one sample. So we took this sample, all of these samples in the field, and we combined them together into one soil sample. Okay? And so this combined, or what we call a composite, we use the word composite, or bulk sample, is then sent to a soil testing lab where nitrogen, phosphorus, potassium, pH, salt content is measured. Okay, but that one sample represents the entire field. From our first video lecture as part of this session 15 lecture series, we discussed how fields are not uniform but have variability across the field. We talked about how soil properties are variable across the field. So when we do a traditional soil sample and we composite these samples together into one sample, the result is we end up with one field or one value for the entire field, even though we know that it is different. Okay? For example, let's talk about a lime and soil pH. In reality, this area of the field could maybe have a pH of 4.5, and this area of the field might have a pH of 6.5, and this in the middle of the field is a pH of 5.5. The average pH is 5.5 for the field. If we apply lime at a uniform rate across the field, this area will get too little lime. This area will get the correct amount of lime. this area will get too much lime. By using a site-specific or a GNS type of soil sampling, we can then actually apply the correct amount of fertilizer or lime in the correct place. One of these approaches to using Global Positioning System, or GNSS, Global Navigation Satellite Systems, to site-specifically soil sample is called zone sampling. Zone sampling. What we do is we identify areas in the field that have similar soil properties. This might be based off of things like yield, soil type, elevation, slope, farmer knowledge. Sometimes farmers know areas of the field which do better than others. And we can then create zones. In this picture, you can see in this map, they have created five different zones for the field. We have one area here, zone 1. So we have zone 1. We have zone 2. Okay, these are actually separated by zone 1, but they have similar properties, so they have created zone 2. We have zone 3 down here, zone 4 over here, and zone 5 down here. And then the farmer or the advisor goes out and soil samples each of these zones separately. So in this example, we would end up with 5 composite samples, or one soil sample, or one composite soil sample from each of the zones. This allows us now to manage the field based on some of this variability that we have. Okay. A second type of GNS, or site-specific soil sampling, site-specific is called grid soil sampling. In grid soil sampling, we create a grid of cells across the field, as you can see in this image. They have a grid of 1, 2, 3, 4, 5, 6, about 7 by 15. Okay, so 7 by 15, and they go and then we soil sample within each of these grids. Okay, in order to create this grid, we have to use a GIS system, and in order to navigate out to each of these soil samples, we will use a GNS. Okay, and so we use the GIS to create the grid. We use the GNS to find the location within the field, and we soil sample at each of these grids. There are some different strategies for how we soil sample at each grid. In some places, we take multiple samples within the cell. That's called grid cell sampling. Another approach is point sampling, where we go to the center of the grid and then just soil sample around the center. Both methods have advantages and disadvantages, but both will improve our understanding of the variability that's in the field. Also, these grids can look differently. They can be evenly spaced, as you can see here, or they can be what we call staggered. Okay, where they're more equal in distance from each other, or they can be random. Okay, most common grids are going to be even spacing, or staggered are the two common ways to go that we soil sample on a grid. After we have collected the soil samples and had the data analyzed in the lab, we can create maps like you see here, where we know the change in pH through the field, so we now can measure that soil pH variability and apply lime based on that. As you can see in this field, lime is not needed in a lot of the areas of the field, but is needed in some locations. Okay? Same with phosphorus, potassium, and we even measured the differences in soil organic matter across the field. In order to create maps like you see here, we will have to use a GIS, or Geographic Information System, and its ability to interpolate the data to create what we call a coverage map. A coverage map. So what are the advantages and disadvantages of these types of systems? One of let's talk about each system as we go through. So let's talk about each of the different types of soil sampling techniques that we discussed in this video lecture. The first one we discussed was traditional methods. Remember, in a traditional method, we take multiple soil samples from within the field, but then we combine them into what we call a composite sample. This system definitely has an advantage in that it requires less labor and is less expensive. In this system, for example, we only will have one soil sample to analyze from the field. But the disadvantage of this system is we do not capture the spatial variability. Instead, we treat the field as one whole. Therefore, it is not accurate for most of the field. Much of the field will have a recommendation higher than what's needed or less than what is needed. Very few areas will get the correct amount of inputs. In zone soil sampling, we do increase our labor costs as compared to the traditional method, but we only create approximately 4 or 5 additional soil samples. So in general, the cost is still low for both the labor and the expense of cost. We do capture some of the spatial variability, but we don't capture all of the spatial variability, and zone sampling will be more expensive than traditional sampling. But it does have the advantage of capturing some of the spatial variability and does an overall good job. The final one is grid sampling. Grid sampling captures most of the spatial variability, and it is a very accurate method for determining that variability within the field and developing sampling schemes or recommendation maps for applying fertilizer and other inputs. But grid sampling is expensive. In this example, just to give you an idea, traditional sampling is one soil sample. Zone sampling could be 4 to 5. But in grid sampling, these could be from 20 to 100 samples, which will be more expensive and more labor intensive. It also requires software to interpret the data and create maps. But the advantage of soil sampling and site-specific soil sampling, that which we do in zone sampling and grid sampling, is we have the potential for reducing soil degradation because we will be applying the correct input based on the sample results to the correct area. For example, if we go back to our original map here, we mentioned the changes in soil pH. Traditional sampling does not measure those changes in soil pH. As a result, this area received insufficient lime and this area received too much lime. The result is this area still has soil acidification. This area of the field now has soil alkalinization, and that can also result in erosion because crops won't grow as well here, as well as over application of fertilizers resulting in nutrient leaching and non-point pollution. Whereas zone sampling and grid sampling will potentially meet those site-specific needs, resulting in more lime being applied here, less lime being applied here, and no lime being applied here. Therefore, the crop will grow more robust, have less potential of soil erosion, and we will not have concerns with soil acidification and soil alkalinization. This concludes our second video lecture from our lecture on applications of GIS and GNSS in agriculture. This lecture we focused on soil sampling.